Thank you, Leslie. Leslie mentioned that I did my college work at Auburn, and I always joked that I paid Auburn so much money they need to pay me some for a while. So I have been with Auburn a long time. I initially started with Auburn primarily doing a whole variety of research projects, from artificial reef fish counts to a lot of shellfish projects dealing with shrimp and oysters, restoration projects, aquaculture. In Alabama, we had some attempts at oyster aquaculture in the early 90s. We grew great oysters, but the gear available at that time just wasn't conducive to off-bottom culture of oysters. So that kind of really took off a lot later. So for today's talk, just give you a quick overview. I'll give you a little background on our hatchery and our laboratory and what we do. Then we'll go into our hatchery production. We'll talk about our containerized spawning system. It's just a way to separate out your brood stock and spawn them without worrying about contamination. Then we'll look at our production systems, our larval production systems. They're batch culture, static culture systems. We are unusual in that we do not grow any algae to feed our shellfish. We use reed mariculture's shellfish diet to feed all our oyster larvae. And we'll discuss how we use that and why we use it. Then in 2015, 2016, we had some production issues. And we're not sure exactly what the problems were, but it happened to coincide with when we were expanding our hatchery. We were designed to be a research hatchery, and we expanded up to a much bigger facility for production to meet the seed demands for the oyster aquaculture industry. When we did that expansion, we started running into some problems, and I'll talk about the problems and how we solved those problems. And then I'm going to show you our setting systems and how we optimize that setting system to really generate a lot of single seed for the oyster industry. So real quickly, we're down here. Our laboratories right here on the east end of Dauphin Island, just off the coast of Alabama. The hatchery was initially built in 2003. We had been doing a lot of shellfish work prior to that. We used borrowed facilities at our Marine Resources Division's facility down in Gulf Shores, Alabama, just using any tanks we could find. But we built this facility primarily to do practical research for the shellfish industry. And with the laboratory, we also built a small-scale hatchery. It was originally designed just to do small batches of oyster larvae and post-set spat to supply to research projects. Never was intended to be a commercial production facility. Well, this guy came to us in 2009. Some of y'all may have met him, Dr. Bill Walton. When he came in and joined us, he brought kind of a renewed interest in oyster aquaculture. And we had one private individual in Alabama that was really interested in off-bottom culture of oysters. So we worked with him and worked with Bill. And Bill brought with him some of the new gear types that were available. And so we tested 4 different gear types to see what would work best in Alabama. And we found out that we could grow really nice oysters. We could control the fouling on the oysters with some of these new gear types. So in 2012, we established an oyster aquaculture training program. Auburn pre-permitted a 65-acre lease. Folks that join the training program will get a little bit of classroom work to learn the biology of oysters and learn all the gear types and best management practices. And then we would throw them out in the field with a run of gear, 10 to 20,000 oysters, and let them give it a try with very low cost. They paid, I think, $250 just to join the program. That got them a run of gear, 20,000 oysters that were theirs to take at the end of the program. If they went through the program, then they had the option to lease 2-acre plots that we had already permitted. So it was a very easy way to get into the business without having to deal with all the headaches of permitting. So now in 2017, we've got 15 operating oyster farms. And these range from folks that are doing 40, 50,000 oysters a year to folks that are doing 2 to 3 million oysters a year. So some mom and pop operations all the way up to some very large operations. And I think we're just scratching the surface here. I believe by the end of this next year, we'll probably be closer to 20 farms. So it's a continuing growing industry. You know, it's growing up in the panhandle of Florida now. Mississippi has joined the game. We actually trained their first batch of farmers this year. Louisiana's getting into off-bottom culture. So it's an expanding industry. So for years, you know, from 2003 to 2009, 10, we were just doing small productions just to meet research needs. Then we started this training program, and we needed a little more larvae, a little more seed. And then the industry took off, and we really had to ramp up production. This is our larval production. The seed production will give you a better idea of what's going on. So again, very small batches of seed just for research. Training program 2012, the first farms coming online needing seed. Some existing farms, the second offering of the training course. So in 2014, we hit a peak of about 12.5 million seed that we supplied to the commercial industry. Well, that's when we realized we got to get bigger. So we went from using 1,000-liter tanks to using 4,000-liter tanks, scaling up our filter systems, and what we did was ruin everything. We had declining production, so we started making these changes in 2015. First part of the season was great. We switched over to all this new gear. Things started going downhill. So 2015, 2016, we really struggled to produce seed. 2016, we did 26 spawns, produced 40 million eyed larvae, and out of that, we only got 7.5 million seed to the industry. We met all our needs in Alabama, but we didn't have anything to give to Florida, Mississippi, Louisiana. We worked our tails off, and normally we start spawning in May, and we usually are done by September. That year we worked on into November. We weren't done getting seed out until December. So everybody worked hard, but it was for such little seed we were getting to the industry. So we were trying to figure things out and trying to figure out what was going on, and we'll look at some of that as we go through. But we obviously figured things out. This past year we did 42 million seed. We filled all our orders, and we supplied an extra 8 to 9 million seed to the industry. So it was a really good year. Oh, and we finished early. We had everybody home for Thanksgiving with no seed left in the hatchery. Just to give you an idea of what we're producing for the industry, and we produce a lot of stuff for research still, and we do restoration projects. But this just looks strictly at commercial seed for off-bottom culture. Majority of it is triploids. We have a few operators that kind of split their crops and do half diploids, half triploids. But probably the primary reason we do so many triploids is we send a lot of larvae and a lot of seed to Florida, and we send a lot of larvae to South Carolina. And the only product we can ship is triploids. We can't ship any diploids to Florida. And for South Carolina, all we can ship is triploid larvae. Can't even ship seed up there. So a lot of our production is geared towards triploids. And we'll talk about that a little bit also. So some of the products we produce, we supply eyed larvae to nurseries, hatcheries. Gave John Gill some this year. We sent some here. We've sent some up to South Carolina, Louisiana. We used to do seed oysters any size anybody wanted. Well, we wanted to evolve the industry, so we had our farms. So now we wanted to get nurseries going. We had a few people that are interested in starting nurseries. So a few years ago, we limited what we would sell to 2mm oysters, retained on a 2mm screen. That would allow the nurseries to take it from there and raise it bigger for the industry. And that would reduce our workload also. We were getting so backed up trying to meet orders that the research was really taking a hit. But we still produce a lot of seed for research projects, a lot of larger seed. We do spat on shell for some restoration work. And we do raise some larger oysters for a variety of different reasons, usually for our own research or other agencies or universities' work. So this year, we've got 3 to 4 folks that are interested in starting commercial hatcheries. One of those individuals said, we can't compete with the university. We would like you to back out of commercial sales. So we've agreed to do that. But what we don't want to do is leave the industry hanging if these new hatcheries don't get online and start producing immediately. So we've agreed to back them up. We'll let these commercial facilities start, take all the orders, but we'll be operating in the background. If they run into trouble or are not meeting what they need for their sales, we'll back them up. We're going to stay in production. We're just going to go into the year like we normally do, stay in production, and if they need us, we're already up and running. We've kind of set a sunset on that. We hope to be out of the commercial business in 2 to 3 years. We'll see how that goes. I may be dreaming, but I would like to be out of the commercial business. So I'll take you through some of our hatchery procedures now. This is something we developed years ago, containerized spawning system. I was talking to Dr. Walton one day, and I said, you know, we really need to write that up. We really need to write a paper on it. He's like, no, it's an off-the-shelf thing you can get for zebrafish. You don't need to do that. Well, now there's a lot of folks that are using this system now. So you've got these 3-liter containers, and each broodstock oyster has its own container with its own water supply. It's a 2-tiered system, and there's containers on the front and the back. So in total, we've got 96 individual units there. So we can run 96 broodstock oysters through there at a time. So in any particular spawn, we can get, in the middle of summer, which is our low point, we get 250 to 500 million eggs out of a spawn. Our peak spawn has been about 2 billion eggs. Our peak actual fertilization of eggs has been right around 1 billion, which is far more than we can stock in our tanks. So the system works really, really well. In the background there's 2. These are 4,000, excuse me. These are 4,000 liter tanks. One of them is a cool reservoir, and the other one just peeking out back there, that's a warm reservoir. So we temperature cycle the oysters to get them to spawn. The heated reservoir, we just got a big titanium heater that we put in there. Heat that water up to 35 degrees Celsius. The cool reservoir, we just blow a lot of air into that water and drive off any heat that's in it. And we usually can cool it down 3 or 4 degrees Celsius just by blowing air into it. So we target about a 7 to 10 degree Celsius temperature change. All right, so we alternate the warm and cool water about on 30-minute intervals. This alone will get them to spawn. But typically, we're doing mated crosses, either single parent mated crosses, or we're trying to develop broodstock lines or producing triploids. And you want everything to spawn a little more synchronously. So what we do is we strip sperm from a male oyster, suspend it in a little seawater in a beaker, and we microwave it. We microwave it in real short increments, 15 to 20 seconds. You want to microwave it just enough to kill it, but not denature whatever chemicals are in there that cue the oysters to spawn. So then we pipette that into each of these individual containers. So every time we change temperature, we'll pipette a little more denatured sperm in there. John Supan coined the term spermescence. So we add that in there, and that gets them to spawn fairly rapidly. You're probably going to have everything that's going to spawn is going to spawn within about an hour. As they spawn, we label the containers, and we put a time on it. So the blue are the males. In this case, we usually use pink for the females, but in this case, it was yellow. We write the time on there because we really want to keep track of how old a particular set of eggs is. We want to make sure we try to fertilize those eggs within an hour of spawning. Okay, so once they've spawned, we take our eggs, we pour them through a 100 micron screen or a 75 micron screen, something that's going to catch any material that's not eggs. So those oysters are in there feeding and pooping in the containers, and you don't want that all over your eggs. So we screen the eggs through a screen, and a lot of times, especially if it's a diploid spawn, we just screen them right into a bucket, a 5-gallon bucket. But if we're limited on sperm, or we're producing triploids where we want those eggs really concentrated when we fertilize them, we'll put a catch screen under there and catch them on a 10 or 20 micron screen. The critical thing with that is those eggs will squeeze through those screens if you just pour them directly on there. If you put them in a little tray of water with a little water over the screen, when you pour them through there, they tend to kind of settle out on the screen. You're still going to lose a few through that screen, but that's okay because especially when you're trying to produce triploids, you want the biggest, best eggs. And those are the ones that are going to retain on the screen. Whenever we're making tetraploids, we also screen the eggs because we want the biggest eggs that can hold all that DNA material. So once we've got our eggs, we like to fertilize within an hour. After an hour, the egg starts degrading and you just don't get as good a fertilization. So if we see stuff start spawning, even if it's not all done, we'll take those eggs that spawn first, go ahead and pull those out and do whatever matings we need to do. So one of the, we really are stringent about what we stock our tanks with. So once we fertilize our eggs, we take 100 microliter samples. When you take that sample, it looks like just a drop on a microscope slide. And we count the fertilized eggs. We count all the eggs and then we count the fertilized eggs. So we get a % fertilization on the eggs. For a commercial hatchery, I probably wouldn't go to all that trouble. All I want to know is the number of fertilized eggs I've got, because I want to know exactly what I'm stocking into my tanks. I want to make efficient use of the space we've got, but I also don't want to overstock it and cause some bad bacterial explosion or not feed them enough. So we're pretty stringent about what we stock our tanks with. Some tricks with triploids. So with triploids, you're going to be mating a diploid egg with a tetraploid male. The tetraploid males, we source ahead of time. We open the oyster, we take a small sample, run it through a flow cytometer just to make absolutely sure it's a tetraploid. So we got the males identified before we start the spawn. So as soon as we get the oysters to start spawning, we start stripping the sperm out of those male oysters, because we want that sperm ready so we can fertilize those eggs as quickly as we can. And again, we really concentrate the eggs. Because typically here lately, the tetraploids we've been using, they're small. They're usually not really ripe. So we want to make efficient use of the sperm we get out of those. We're not getting much, so we try to concentrate those eggs down to as small volume as we can. And then when you add the sperm to them, once you see that everything's fertilizing properly, then you can fill it with some filtered seawater and get a larger volume. With triploids, we overstock our tanks. So normally we may stock a tank at 10 larvae per milliliter. If we're doing triploids, we'll stock them at 20 larvae per milliliter. We expect losses with triploids. They just don't survive as well as diploids from fertilization to the first drain down 2 days later. We expect losses. So we overstock our tanks because we have a high demand for triploids and we want to get every one of them we can. But about as high as I've ever gone is double. It's about 20 per milliliter. So in the early days, these are the tanks we use. These are 1,000-liter tanks, about 250 gallons. This is what we used for 12 years. Did great for small research batches. I could get anywhere from 1.5 million to 3 million larvae out of one of these tanks to set. Again, we stock them at 10 eggs per milliliter, and that's fertilized eggs. We don't worry about the ones that aren't fertilized. So we stock these at about 10 million a tank. And then through the course of the larval cycle, we lower those densities down to where at the end we're harvesting about 3 million a tank. 3 million would be about the maximum you're going to get out of one of these tanks. You know, realistically, you can expect 1.5 million to 2 million. So in 2015, when we decided to kind of scale things up and ruin our whole process, we put in these 1,000-gallon tanks, or 4,000 liters. I love the tanks. I love the setup. These tanks have a slight cone to the bottom. The previous tanks, they have a real deep cone to them. These have very slight cone. We just want just enough slope so that when we're draining the larvae, everything drains down. But you don't get all the sludge sliding down. Again, stock them at 10 eggs per milliliter, so about 40 million in one of these tanks. So one of these tanks is equal to 4 of those other blue tanks. And we hope to get about 12 million larvae out of one of these tanks. Realistically, we use 6 of these tanks at a time for any particular batch. And we're probably the best we've done is about 30 to 35 million out of 6 tanks. So this is a theoretical number, but we end up, you know, towards the end of the spawn or end of larval rearing, we throw away the little ones that don't quite make it. So you're looking probably more like, you know, 6 to 8 million a tank instead of 12. But once we got this working, this works great. We love it. The tank has a center standpipe, and then it's got a 4-inch sleeve over the top of that. And then at the very bottom, there's air lines that enter on either side of that pipe. And if you can see at the bottom, there's small little scallops cut in the bottom to allow water to flow in there. So we inject the air down there low, carries that water up to the top, and creates this nice, gentle circulation through the tank. And that's important when you're using shellfish diet or an algae concentrate. It's not live algae, so it doesn't want to stay in the water column. Over time, it wants to settle out. Well, if we create that nice flow, it stays suspended up in the water column really well. We also don't use any air stones on this. These are just air lines directly into the side of that pipe. We want really big bubbles in there. One, it pushes a lot of water, but when you're using the algae concentrate, it has a tendency to want to foam, and it's like a protein skimmer. you put a lot of air bubbles or a lot of small air bubbles with air stones, it wants to foam. With big bubbles, less surface area, it doesn't foam. So that's why we're not using any air stones. But just that gentle flow is enough to keep those larvae in suspension, the algae in suspension, and everything aerated really well. This is something we did. We actually started doing this in 2016. We noticed we were getting little patches of algae settling out. And those little patches of algae would tend to grow bacteria on the bottom of the tanks. So this is something we did to keep that algae in suspension, and the stuff that does settle settles in a more even coat around the tank. Intensity of aeration. We use very, very gentle aeration. So let me go back a slide. So basically, it's going to create some ripples as that water's coming up. But the way I gauge it is if those ripples hit the edge of the tank, that's too much aeration. I like it to kind of die down before it hits the edge of the tank. So it's very gentle aeration. I coddle my larvae. I like to treat them very gently. So water that we use for raising the larvae comes from these two storage tanks. We fill these with raw water straight out of the Gulf of Mexico. Our salinity varies quite a bit. We've had times when we can go fresh. We're pumping straight out of the Gulf of Mexico, right off the front beach, but we're right outside the mouth of Mobile Bay. All the fresh water that comes through Mobile Bay hangs right in front of our hatchery. So there are times when we have a lot of water coming down the bay that we have to target filling our storage tanks on a high tide to make sure we're getting good salinity. I like salinity above 15 parts per thousand, 15 or above. I'm happy with 12 and above. I can make 10 and above work. But I much prefer a little higher salinity. If it's 10 parts per thousand and above, it generally works pretty well. You get below 10, the larvae are not happy. You get below 5, then they're toast. I'll make a point about filled with raw water. We're going to come back to that in just a minute. We don't do any pre-filtration on this. So it's just raw water pumped in there. It gets a chance to settle for a day. One of these is enough to do a water change on 6 of our tanks. So we'll use one tank one day, refill it. It gets to settle for a day while we're using the other tank the next day. And I'll mention that we use 6 larval tanks at a time. We actually have 12 in the hatchery. We could raise a lot more larvae in those 12 tanks, but it becomes very cumbersome to try to drain a tank, clean it, get it refilled, and get larvae back in it. So we like to have a completely clean set of tanks ready and waiting that we can be filling as we're draining tanks. So when we drain our tanks, we do our health assessments, and then they can go straight into the new tanks. Again, just try to treat them like your children, you know. Keep them happy. The water goes through a filter system. We filter down to 1 micron, and we go through a series of cartridge filters and bag filters. And you also see a UV sterilizer here. We'll talk about that some more a little later. It's a 2-sided system, so there's a cartridge filter here that's 10 microns, a bag filter that's 10 microns, a bag filter that's 1 micron, and then there's a whole nother set here. It's valved so that you can shut one side off, take the filters out, clean them if you need to, put them back in, turn that side on, clean the other side. Typically, we don't have to do that. Typically, we run them for a day, take them out at the end of the day, hose them out, let them dry out overnight. We're ready to go for the next day. I know some people take those bag filters and bleach them. We're very conservative with bleach. We don't use a lot of bleach. We do use bleach to clean tanks. But But when it comes to filters, we just hose them out with fresh water at the end of each day. All this is on a pallet. Hurricanes. None of us are immune to them. So you got to be able to get this stuff either moved up to high ground or loaded on a truck and get it out of there. So everything is valved with these union valves. You can break those apart. The pallets will separate, the components will separate, and you can take each of those pallets, load it on a cart. We just take them upstairs. The hatchery itself is about 7 feet above sea level, and if we get them upstairs, we're another 10 feet above sea level. We've had a couple of big storms. We had Hurricane Ivan in 2004 and Hurricane Katrina in 2005. We had a different configuration to the hatchery, and we actually had all our nursery in the same area also. Those storms took all those tanks and just kind of pushed them to the back of the hatchery. Never turned any tanks over. We never lost any animals. All the tanks stayed upright. They were just in a big pile in the back of the hatchery. But you want to make everything easy for you when a storm's coming. You want to be able to break this stuff apart and get it to high ground as quick as you can, because you've got other things to tend to. You've got your own home. So we try to make everything as quick as possible when we got a storm coming. Just a different shot from the other end. So our water's coming in from somewhere from over here, comes into the pump here. There's a bypass here. If you don't want to go through the filter, if you just need some water for something that you don't want filtered, you can bypass it here. Again, you can shut off one side if you need to clean one set of filters. Again, these are cartridge filters. They are adequate. These are actually pool filters that you get from Pentair. They are adequate. We've used them for years. They're fine. I'm actually looking at Hayward filters now that are designed for more heavy-duty use. These have a plastic insert in them that's a little flimsy. This locking ring here, it goes bad on you sometimes. I love these bag filters. These are heavy-duty. The threads on the lids are really thick. They don't tend to strip. I really like these. These are a Pentair product. But I'm looking for something a little more heavy duty here. It's adequate. It's lasted for, I guess we've been using those for 3 years, and they're still good. But they do have some wear and tear on this locking ring, and the components inside are not heavy duty. I'm going to try to go to something a little heavier duty for this. All right, we kind of locked up. There we go. All right, draining tanks. A lot of you probably seen folks that use just big cylinders to drain, open top cylinders, drain right into it on a screen that flows out the bottom. Several years ago, actually pretty much as soon as we started raising larvae in the hatchery, we went to these bag filters. These are a bag filter that you would use in a canister filter. They use them for a lot of different things. But the great thing about them is you can tie them on here. You can tie them tight enough that you can walk away from that tank, and if you get a phone call or a text message or your major professor calls you away, there's not going to be a disaster here. With an open top screen, if you get distracted and that screen clogs and your larvae spill over, you're starting to lose larvae. We work with a lot of interns, a lot of graduate students, and they all get distracted very easy. So we started using these bag filters, and I love them. Once we drain the tank, we can rinse them a little bit in that bag filter to get rid of some of the excess algae, and then we just rinse them down to the bottom, invert it into a 5-gallon bucket. At that point, we raise the volume of that bucket to like 10 liters, something that's really easy to calculate numbers from. We'll take samples, look at them under the microscope, count them, assess the health. and then we put them back in a new tank of water as quickly as we can. But I love this. This is one trick that I think it's great. I can't tell you how many times I saw screens overflow over the years, and this has eliminated that. Okay, so we stock initially at 10 larvae per milliliter. Over the course of our larval rearing cycle, we reduce those densities down. Some of that's just natural attrition. You're going to lose some. There's some that are just not going to make it. But we count, every time we do a drain down, we count our larvae to see how many we've got and how many we're putting back. If we want to reduce densities, a lot of times we will screen them. We'll use a slightly bigger screen than they screened on the drain down before, catch the bigger larvae, and if we need to reduce densities, we'll just flush the small ones. Okay, so kind of our density reduction schedule. Again, day 0, 10 per milliliter. So that very first drain down, our first drain down is day 2. We drain every other day until about day 8. So our first drain down, we cut that density in half right then and there. Hopefully we've got enough tanks that we can spread those out. If we don't, we'll drop these on like a 40 micron screen or something. Anything that goes through, we'll just let those go and keep the bigger, really nice larvae. And also the screening, you can see in a sample like this, you got some little guys here, some stuff that's a little smaller, and then some stuff that's a little bigger. You can also, screen off the bigger stuff, put them in their own tank, put the little ones in their own tank. They'll grow better if you keep like sizes together. Day 6, we go down to 4 per milliliter. A lot of this, you get to these densities just through natural attrition. But if you don't, you can use those screens to lower those densities. Day 10, we like to be down to 3 per milliliter. Day 10, we're usually harvesting our first eyed larvae. It's just a trickle on day 10. We may get a few 100,000 on day 10. Day 11, we'll get a million or two. Day 12, we're 2 to 3 million. So we kind of hit a peak around day 12, 13, 14. By day 16, we're headed down the other side of the slope, and the numbers are dropping off. So we get these really big collections of eyed larvae, right there between day 12 and day 16. It's kind of our sweet spot. So when we first start harvesting eyed larvae, we may not have enough to really justify setting at that point. So we may stick them in the refrigerator. You take your larvae, put them on a Nitex screen, wrap them in some saltwater wet paper towel, throw them in the refrigerator. They're going to be fine for a day or two. Then when we get more larvae the next day, then we can set a bigger batch of larvae all at once. So again, we start screening for eyed larvae on day 10. I've seen them as early as day 9. That's rare. But typically, if we got a good healthy batch, we're going to start pulling the first few eyed larvae off at day 10. When you start seeing that eye spot, they're getting close to ready to set. For diploids, we screen on a 200 micron screen, and for triploids, a 224 micron screen. You may talk to other hatcheries, and they may use slightly bigger screens. I've tried going to bigger screens. I've tried using a 210 here and a 230 here. But what I found is I start losing a lot of larvae that set to the insides of our culture tanks. They're growing so fast at this point that if I leave them just another 12 hours, they start setting on standpipes, they start setting on sides of the tank, and it's easy to lose several 100,000 or 1 million larvae that set inside the tank, inside of your culture tanks. So we found these screens to kind of be optimal for us. The larvae that come out are maybe not quite as hot as if we went to a bigger screen. So we give our larvae a little longer to set. We usually give them 72 hours to set once we harvest them. If your larvae are really hot to set, usually 48 hours is long enough. So I mentioned we use reed mariculture shellfish diet. Most commercial hatcheries, most hatcheries in general, raise their own algae. When we first started in the new laboratory, we started using this product. Well, actually, we had been using it for years before and had good success with it. Not many other people were using it, and anybody that did use it just said, oh, that stuff doesn't work. It worked for us. We've got a pretty good relationship with Reed Mariculture. I spent a lot of time on the phone with their lead scientist, Eric Henry, and he's given me some tips over the years on how to get it mixed up right and use it. So over the years, we've come up with a set of protocols. If you got this sheet on your way in, there's some directions in there about how to mix algae, and we'll talk about some of that. But it's really worked well for us. So why did we decide to use this? We have a small algae lab. We have a small space dedicated to raising live algae. But it's not big enough for the quantity we need for the volume we're doing now. But not only do we have space limitations, we got staffing limitations. We have 3 full-time staff. We've got 2 farms, research projects, graduate students to deal with, a hatchery and laboratory to maintain, and research projects to run, and hatchery production to do. We really don't have staff to dedicate to raising algae. And it works out cost-wise. When you take into account the equipment you need to raise algae, the chemicals and supplies you need to raise algae, the personnel, the commitment of space. Last year we did 42 million seed. We did 180 million eyed larvae. We spent maybe $10,000 on algae. That would just be, a fraction of one person's labor to raise algae. So it works out for us cost-wise. The one caveat I will say is that this is the only product I've found so far that works. And if something ever happens to them, I'm in trouble. We're going to be in the algae growing business really quick. In fact, I want to talk to them about their succession plan. The Reed guys are my age. They're getting older. They got to start thinking about retirement. I want to make sure they're going to keep growing this product. So this is a composition of several different species of algae. It's centrifuged down and it's mixed in different, the different species are mixed to make that diet. It's Isochrysis, Pavlova, Thalassiocera, Tetraselmis. I believe that's all of them. They may have added one more recently. There may be 2 species of the Thalassiocera. Some of the cells in there are too big for the first larvae, for the really small size larvae. So we've played with it to try mixing different species, some of the smaller cells for early larval stages. But we've yet to come up with a better mix that works than this. This works really well. We've been very pleased with it. This looks confusing. It's really not that confusing. There's on this sheet, lower right corner, there's some feed rates there. Those feed rates worked fine for us for years. And I've got former students that are still using these feed rates on this sheet. But when we started having trouble a couple of years ago, one of the things we looked at was our feed rates. And I decided that maybe we needed to feed a little more early in the life cycle. So this is a new spreadsheet we've set up for calculating feed rates. It's fairly straightforward once you understand it. And I'm happy to share this with anybody. I'll give you an email address here in a second, and you can just email me. I'll send it to you. So you've got your day over here, your cell density. This is the target cell density we're shooting for to maintain in the water column. If you read some of the hatchery manuals, these numbers will look pretty low. But reading through the literature, I found that when you put a whole bunch of algae in there at one time, the larvae don't feed real efficiently. There's kind of a sweet spot for cell density. If you can maintain that cell density, it's even better. So we've got our larval density here. And just reading through the literature, I came up with some ingestion rates, how much algae each larvae is consuming. So you take your cell density that you want to maintain, how much they're eating, and you get these different calculations. So when we do a drain down, we need to add some algae to that tank. It's going to get it to that right cell density that we need. So that's what this column is, initial feed. And this is for a 1,000-gallon tank. You can take this spreadsheet and change the formulas in these blocks to fit whatever tank size you've got. It's just changing one number, the volume of the tank in milliliters. So this is what you want to put in the tank to get that cell density up to these numbers here. Okay? We use a continuous feed system. We mix our algae, put it in a refrigerator. There's peristaltic pumps, I'll show you that in a minute, that take it to our tanks. So this column gives you the amount of algae that you need to put into your feed system. So it's the amount of algae of that concentrate that you use. Depending on the feed rate of your peristaltic pumps, you'll have to calculate for yourself what kind of volume you need to mix that in to last 24 hours. But this is the volume of shellfish diet you need to mix, put in your refrigerator, and feed to your tanks over the next 24 hours. I mentioned that for the first 8 days, we drain every other day. So day 1, 3, 5, 7, we're not doing drain down, so we're not feeding this part. I'll actually, early in the life cycle, I'll just add this to this number over here. And it's just a little extra boost. If you don't want to deal with a continuous feed system, we found that batch feeding twice a day works pretty well. It's a lot less hassle. We get very similar growth rates. Just intuitively, I feel like they're feeding better if I can just keep that cell density constant throughout the whole day. But batch feeding twice a day works pretty well. So you can just take this number and you're going to feed that amount twice a day. And this is milliliters of shellfish diet. Okay, so if you want to copy that down, that's my email address. If you want a copy of that spreadsheet, I'm more than happy to send it to you. It's a work in progress. What I hope to do is develop it so that you can just tell me how many larvae you got, tell me what size they are, tell me what day they're at, and it'll spit out a number for you. I'm trying to make it more automatic instead of having to calculate it all. But I'm going to say this, there's no cookbook recipe for raising larvae. There's a lot of art to it. So if you see that your larvae are lagging behind a little bit, so say they're at day 8, but their size says they're at day 7, well, just drop back a level on that feed. If they're not consuming all that algae, just cut back a little bit. There's a lot of intuition and art to it. If you stick strictly to a recipe, you got to give yourself a little room there to massage it a little bit. But again, I'm happy to share that spreadsheet with anybody. Mixing the algae. So you've got your volume from that spreadsheet that you need to mix up. We initially mix it in a pure sodium chloride solution. So that's just uniodized salt mixed in fresh water. Mix it at 30 parts per thousand. So that's 30 grams per liter, roughly. We mix that solution up. We mix 20 liters at a time and keep it in a carboy. And we mix our algae with 500 milliliters of it. what this does is it disperses those cells. If you drop that algae concentrate straight into seawater that's got a lot of organics in it, it's going to want to clump up. All those organics just make it want to just stick all together. So what this step does is just breaks those cells up, disperses those cells. Once you've done that, then you can mix it in. You can dilute it with some 1 micron filtered seawater. At that point, it's going to spread out. We also pour it through a 20 micron screen. Again, that just further breaks those cells apart and gets it real dilute. Okay, so we mentioned 2 feeding regimes, batch feed or continuous feed. For batch feed, you're going to split your ration into 2 batches a day. So we feed them first thing when we come in the morning, last thing before we go home at night. And typically we just pour it straight through the screen into the tank. For continuous feed, it's actually a very simple system. We mix our algae in a 5-gallon bucket. We fill it to a volume that we know how much these are going to pump. We fill it to a volume that we know is going to last 24 hours. It's a tiny little circulator pump. This is something you get from an aquarium supply store. And it's got a little suction cup. You stick it on the bottom and it just shoots water around the bottom of that bucket. We don't put air on this. If you put air on it, it's going to foam. It's going to foam and create a mess. A lot of it will stick to the sides. There'll be a lot of it that never makes it to your culture tanks. Okay? So these little lines here, these little tubes, feed these peristaltic pumps. Okay? This peristaltic pump is plugged into a cycle timer. I love these. These are called a raindrop cycle timer. It's hard to find them these days. You can find much more expensive versions of this. This is I don't know what I'm trying to say. It's not a high dollar piece of equipment. This is like $5 for a little cycle timer. It's got little punch tabs that you tell it how long you want it to be on, how long you want it to be off. This runs on 10 minutes off, 2 minutes on. So it's going to run 5 times an hour. Okay? Each time it runs, it's running about 14 milliliters of algae per minute. So each time it's on, it's running 2 minutes, so it's doing 28 milliliters of algae. Doses it right into the tank. The outlet line on it, I just clip it right on the standpipe in the middle of the tank. So when it drops in, it's dropping in right where all that water is moving up from that standpipe. And so it further mixes into the tank from there. Very, very simple system. I wish I could find these timers again. They do tend to wear out over time, but they're so simple and they're so cheap. Most of the stuff you find these days is about $30 a piece. These are $5 a piece. Okay, so this just kind of shows you that the batch feed and continuous feed, they grow about the same. Intuitively, I like the continuous feed idea better. I like the idea that they've always got food there at the right density. I feel like I'm using my food more efficiently. Shaded area, this is where we start pulling off eyed larvae. So even though our average is just below 200, we've already got some stuff that's in the 280 range that we're pulling off. Okay, so let's talk about hatchery problems a little bit. So I mentioned earlier, we had our first record year in 2014, about 12.5 million seed. The next year, we changed over to these bigger tanks, these fancy filter systems, and production started going downhill. 2016, we struggled the whole year trying to figure out what was going on. 2017, we decided, okay, we've got to take a 2-year step back, figure out what was working before, what we're doing different now, and fix it. We made a lot of changes. I can't tell you which change made the difference or what the difference was, but I'm going to tell you what we did. And if we can get out of the commercial business, we're going to spend some time figuring out what the secret sauce is. So what were the problems we were seeing? We'd get great batches of larvae. They do fantastic. We'd have millions of larvae. They'd be growing well. And then day 6, 8, you'd start seeing them feeding real poorly. All the algae wouldn't disappear from the tank. You'd start seeing stuff like this, just a lot of clumped up algae. You'd see empty shells, just not good looking larvae. Slow growth, they'd be days behind where they should be. Day 9 or 10, that's when disaster would hit. We'd see just big clumps of black larvae that would just be sitting on the bottom of the tank. We'd screen them out. We'd try to put them in some clean water, resurrect them. Occasionally we were able to, but very seldom would they come back. Very depressing. You've already got 10 days invested, and then there's all those larvae millions of larvae laying on the bottom of the tank. It was a very, very stressful time. So, we're trying to figure out what the issues were. And one thing we kept coming back to was bacteria. And so we started running some samples with we've got an FDA lab on Dauphin Island. They were willing to run samples for us. Assessing these problems after the fact is very hard. You got to do it while it's happening. And it's very hard to do bacterial samples in real time. But we just do the best we can and try to make some suppositions from the data we get back. So one of the things we saw we were having some Vibrio issues with a few batches. Vibrio alginolyticus. It's been shown to be a problem in some hatcheries. We noticed we had high numbers in there, but we couldn't say for sure that that's what the problem was. Was that the problem, or was that the result? Was that Vibrio moving in after the larvae were in poor shape? But what we did find that was interesting was that our poor performing tanks were dominated by 1 or 2 species of bacteria. And we also saw this in viral assays. It would be bacteriophages. And that's a bacteriophage is indicative that you got a bacterial issue. But our better performing tanks had these really diverse bacterial communities. So the ones that were doing well had these real diverse communities of bacteria. The ones that were doing poorly were dominated by 1 or 2 species. So it got us to thinking, okay, what's allowing those 1 or 2 species to take over? And obviously those 1 or 2 species are causing a problem. So we started thinking about what we had done different. When we scaled up the hatchery, we had a need to really process water a lot faster. We went from filling a few 250-gallon tanks to filling all these 1,000-gallon tanks. So we wanted to filter our water more efficiently. So one thing we did is we put a pre-filter going into our storage tank. So it took all the mud out. We had these vortex filters on here, and then a cartridge filter. So we filtered that water down to 20 microns. So we kept a lot of that mud and sludge out of our tanks. Well, this is the one thing we came back to. We took this system out. So what our thinking here is, we took all this sludge out of our storage tanks. Well, maybe that sludge is the bacterial soup that these guys really need to keep that bacterial population diverse and not let one particular species take over. We felt like we were cleaning our water up so much and getting it so pristine that we were just creating this great environment for one bad bacteria to take over. If we keep the bacterial population real diverse, then nothing gets a foothold. And I think, and we need to do more research on it, but I feel like bacteria is not recognized as a food source. I think that it's a significant food source for larvae. And I think it also helps in their gut. Like, we need our right bacteria in our gut to digest food properly. I think oysters are the same way. And if you eliminate all the bacteria in the water and they're not getting any, then I think we're causing a problem, especially if you get one bad bacteria that takes over. So we took this out. We want that sludge in the bottom of our storage tanks. We just let it build up. We clean our tanks out once a year. This winter, some graduate students got to get in there and clean out the sludge. We also eliminated the use of the UV. I mentioned we have this big UV filter on the filtration system. We don't use it anymore. Again, we're sterilizing that water, killing all the bacteria, the bad bacteria and the good bacteria. And there's always going to be some that get through, and maybe it's the bad bacteria that get through and explode. So we eliminated using the UV filter. We had a big conference call in 2016 with hatchery managers up and down the East Coast, and all of them said, take the UV out of the system. None of us use UV. So, which was counterintuitive to us. We felt like cleaner water, better water. Well, we've kind of gone the other way. Dirtier water, a little more bacteria, a little more food. So we eliminated UV. Other things we did. The new spreadsheet I talked about, it actually increases the feed rates early in the life cycle. You want those larvae as healthy as possible going into the later stages. Again, I can't say for sure that this made a difference. I've got former students and colleagues that are using these feed rates and doing just fine with them. But I feel like our new version works a little better. So we increase the feed rates, especially early in the life cycle. Daily drain downs. We used to do drain downs every other day until we got to the point we were harvesting odd larvae, which was usually around day 10. Well, now we don't push it any further than day 8. After day 8, we go to daily drain downs. Drain the tanks, change the water every day. Again, that's just because we started seeing problems at day 9 and 10. I can't say that this fixed the problem. It may have been the filtration. It may have been not using the UV. This may have nothing to do with it, but it's working. So we're going to stick with it. EDTA, this is a salt. We add it to our tanks at 1 part per million. This was a recommendation from the guys at Reed Mariculture who had looked at Vibrio a lot more than we had. And we're not exactly sure if this is really working or not, but right now everything together is working. We got to take these components apart and test them individually and see what's really making the difference. But 1 part per million EDTA, what it does is it pulls the metals ions out of the water, chelates the metals, which the Vibrio bacteria need to flourish. So again, I can't tell you for certain that that's made a difference, but it's working. Things are working. So we've all heard the disaster stories of the East Coast and West Coast, ocean acidification, problems with pH and aragonite saturation and alkalinity. It's a very hard question to get at. You're in estuarine waters that fluctuate so much. pH is all over the place, alkalinity is all over the place, which makes your aragonite saturation all over the place. Very hard to figure out if this is really a problem for us. We targeted pH at 8.1 to 8.3, alkalinity at 160, and aragonite saturation above 2. We did this in 2016. We would use soda ash to raise pH, bicarbonates to raise alkalinity, and we try to find that sweet spot for aragonite saturation. I will say that we produce some of the most beautiful D-hinge larvae I've ever seen. You know, when you screen those D-hinge larvae and they're pink in color on the screen, these were bright red. I mean, these were gorgeous larvae. But at day 10, they died just like everything else. So we may have improved the early stages. And this is something we got to look at a little more. but it didn't solve the problem. Did not solve the problem. But I do want to come back to this because I do feel like we were getting really good larvae to start with. And if we can improve the larvae we start with, hopefully we improve the product that's coming out the other end. All right, let's talk about setting. So the majority of what we produce for the commercial industry is single seed for the half shell market for off-bottom aquaculture. So we use an upweller system. Actually, it's a downweller system, which is a converted upweller system. And we set it on real finely ground oyster shell, 250, 300 microns. We used to screen all our own culls. This is what students and interns hated the most. It takes a long time to screen crushed oyster shell to get a lot of that particular size range. And then we found a guy on the East Coast, Baywatch shellfish, I believe, Keith Baya. Is that it, John? Okay. He's got a machine that crushes it and sieves it and grades it. And when I can buy 10 pounds of it for like 80 bucks, oh, it saves so much labor. Great thing. If you're setting seed, call Keith. Baywatch shellfish. But we use this specific size because we're setting on a 180 micron screen. We put this material on there, very thin layer. All you want is enough just to cover the screen. You don't want a thick layer because what happens in a thick layer is the interstitial space, that water in between those grains goes anoxic and you start getting some funky things happening. The culch will start turning black on you. So you just want just enough, real thin layer, just enough to cover the screen. And it's sized so that, the larvae itself is about 300 microns. And that piece of culch is 300 microns. So as soon as it sets, you can drop that on a 300 micron screen. Everything that's got oysters will retain, everything that doesn't will fall through. So very quickly you can get rid of that excess culch. We take the excess culch, put it in a drying oven, and we'll set it again. Oftentimes we'll screen it off, put that culch right back in the tank and set it again. So the system. This is our standard upweller tank we use for nurturing seed. For setting, we just reverse the flow and keep very low water in there. It's a recirculating system. What you see over the top there, we put covers over it when we're setting. We like to set them in the dark. So we put a small little mag drive pump in the bottom of the tank, pumps it into the center trough, and that center trough feeds what would be the outlet on an upweller. Now it's the inlet on a downweller. Put a 2-inch cap on there. There's 2 tiny little holes drilled in that just allow a little sprinkle of water in there. Just a little bit of movement. Circulates that water. We feed our larvae when we're setting. Okay? So you get a little bit of circulation. Again, if you just scratch that surface of that culch, there's screen right under it. That's just a very thin layer. So this is a 22-inch silo, and we put 150 milliliters of culch in there. And we found that not only is that culch size great for screening off the spat, but it's also great for producing singles. We found if we go any bigger than 300 microns, we start increasing the number of multiple sets, or we call them doubles, where you get multiple oysters growing together. Right now, we're achieving 95% or better single oysters with this. Typically, we're up in the 97, 98% range where they're single oysters. You can get farmers really crabby by giving them a batch of seed that's full of doubles. I'll make one other mention on that. When we sell our seed, because we are a university, we want everybody to be happy. So when we estimate our seed, we do ours by weight. When we're doing our counts, for weight or sample counts. We don't count empty shells. We don't count doubles. So those are accounted for. So we want to make sure when people order 100,000 oysters, they're getting 100,000 single oysters. They may be getting 105,000 oysters, but the rest of them are doubles. We want to make sure they're getting the single oysters they ordered. The other thing is we add 5%. I don't know if that's a good business model in the commercial industry, but we add 5% to make sure folks get what they ordered. And if they get a little extra, it makes them that much happier. So back to the system, very little water in here. We set the level of the water by filling the tank to where the water level is just 1.5 inches to 2 inches above the surface of that screen. Not only do the oysters like to set on the culch, they like to set on the sides of the silo. And if you can minimize that surface area, you minimize the amount that are going to set on the sides. The other thing we do that we found a few years ago that really helped, typically we were getting set rates of 30, 40%, and pretty happy with that. A lot of folks were happy if they were getting 20%. But if you go to Lowe's and you go to the paint department, they've got a little tool for caulking. It's a little rubber, it looks like a little rubber scraper, a little square piece of rubber that's beveled on the edges. If you take that bevel and you run it right along that edge, the oysters that are set on that side will ride up that bevel and fall off into the screen. If you do this every day before they get a chance to grow, there'll be a lot of them that survive that. You're still going to lose some, but a lot of them will survive that. We increased our set efficiency. This year, we were setting above 60% set rates. So for every 1 million oyster larvae we're putting in there, we're getting 600,000 out. That's really good for setting singles. Really pleased with that. So just a little simple trick that can really help your efficiency. Okay, just a few other things. We soak our microculture at least 1 day ahead of time. Typically, we'll soak it a couple of days. We'll fill a whole tank. We may only set a silo or two, so the rest of it's getting soaked even additional time. We stock the silos at 300,000 larvae per silo. That works out to about 125 larvae per square centimeter. A lot of your manuals will tell you 100 per square centimeter, and that works fine. We've been able to push it up to 125 and still get a lot, the majority singles. Like I say, we're 97, 98% singles most of the time. Another thing we want to look at down the road when we can get back to doing research, we want to see how high can we push this and still maintain our singles production. Because we've got a lot of commercial guys, they're like, well, I want to set 500,000 in there. Well, you can try it. I can't guarantee you that you're going to get all singles. I know this number works really well, and I've been pretty pleased with it. Last year we did 42 million single oysters for the industry. That was 6 times what we had ever done before. No, I take that back. It was 6 times what we did the previous year. It's probably 4 times our record of 2014. So again, I know this number works, but we do want to see if we can push that even further. Water level, again, 1 to 2 inches of water above that culch. Don't give them any more surface area to set on than you can help. Remove the set from the sides daily. You'll really increase that setting efficiency. We do feed our larvae. A lot of people won't feed during setting. They say they're undergoing metamorphosis, so they're not going to feed. Well, you've also got larvae in there that aren't quite ready to set. They're going to be another day or 2 down the road setting. So you want them to keep feeding. After they set, after they metamorphose, they're going to want to feed, and they're going to want to feed a lot. So we put 10 milliliters shellfish diet per 1 million larvae. This works really well, and we do that twice a day. We don't try to do any feed systems on that or anything. It's just a batch feed twice a day. We cover the tanks. They like to set in the dark. We give them a nice dark environment to do it. I will tell you, we do this in a greenhouse. It's just a greenhouse roof, no sides. We lost the roof in a storm. Freak storm came out of nowhere. It wasn't an expected hurricane or anything. We lost the roof. So now we're in the beating sun on these tanks. So you put a nice piece of black plastic across the tank, it heats up quick. So we may leave the ends open just to get air circulation under there. But if you're in an open sun environment, be careful you don't cook your oysters. We allow them 72 hours to set. A lot of the manuals you'll see will say 48 hours. Again, we're harvesting maybe a slightly smaller eyed larvae. And we need to give them just a little bit longer to set. The majority of the larvae are going to set in the first 48 hours. But you can look in there at the size they are. You can look in there, and if you see a whole bunch of little black dots swimming around, give them another 24 hours to set. 72 hours works really well, but you do want to do a water change at 48 hours. Just like the larvae, that water does get fouled over time, and you want to change that water. So we'll quickly drain the tank, scrub it as fast as we can, refill it, and give them another day to set. We sieve off the spat on 300 micron screen. We used to take the silos right out of the setting system, put them in a flow-through system, give them a few days on flow-through before we screen them. To increase efficiency so we could keep setting, we would go ahead and screen off the spat, move them to an upweller, a flow-through upweller, and then take what went through that might still have a few larvae. It might have some spat that didn't set on culch or set on the sides and fell off that aren't retaining that 300. And you also got good aged culch. We throw that right back in the setting tank and reset it. Throw another 300,000 in there. So every 3 days we screen them off, we move them to another tank and reset that. So as long as we got larvae coming off, we want to keep setting them. Also, if you're running a bottle upweller, this works really well. A lot of folks will take all their culch and their spat, throw it in a bottle upweller, let that grow for a while in a bottle upweller, and then screen off the spat. Well, doing it this way, you can put a lot more seed in a bottle upweller if you don't have all that culch in there. Also, all that culch in there is just added friction that's rubbing on those shells. So if you get rid of that, you're left with pure oysters. We're very gentle with them at this point. Our flow-through is just a trickle. I mean, we want just enough flow that they're getting enough food and growing. We don't really put the flow to them until we get them on a 500 micron screen. So for the first week, when they're in an upweller, it's very slow flow, but they are feeding now on what's in the water naturally. So the instant they're set, they're on flow-through water. We're no longer feeding them. We're out of the algae business. Mother Nature's taking care of it from there. They grow really quickly. Usually in 4 days to a week, we're up on a 500 micron screen, and we're hitting them with as much flow as we can pump to them. So I'll leave it at that. That kind of gets you through the Auburn's whole hatchery process. There's a lot more that comes after that we also do, but we're going to leave it at the hatchery stuff for today. But if you got any questions, I'll be happy to take them.
On your drain down bags, what size mesh are you using?
Good question. Yeah. Okay, so the question was, what size are the bags we use for our drain downs? So our first sieving is on a 20 micron bag. After that, all the sievings are done on a 40. We started using a 75 at some point, but the 75 bags have much bigger stitching around the edge, and we noticed we're starting to lose some larvae getting through the holes of the stitching. But the 20 and the 40, these come from Pentair. The 20s and the 40 have felt that's stitched in on the seam, so the larvae can't get through that seam. So that works really well. We will, another little trick is we keep a saltwater hose running all the time. So as they drain down and that bag swells up and you get a little algae clogging, we just spritz it with a little salt water, and that kind of flushes that algae that might stick in the screen through.
Any other questions here?
John?
Well, on that same note, you talked about sieving off the bigger ones to set and saving the smaller ones back to the tank. Do you have a second silo under it, or how do you catch what's not the ones that you want to set?
Sure. So the question was about sieving larvae through multiple screens and how to catch them. So we generally know what target size we're shooting for. So maybe we're using a 75 screen. And a lot of times we'll throw away what goes through. Sometimes we may want to keep them. If we're trying to absolutely maximize production, we'll keep everything. So we'll just put another screen underneath it. Just a catch screen of 40 microns or something.
You sieve the setters through something different than
Okay. If you okay. So the question is our setters. What do we do with the larvae that go through on a sieving screen for harvesting eyed larvae? So again, we just sieve them in a tray, and either we'll have a screen under it, or we'll just sieve them in a tray and then pour that tray into another screen to catch those. A lot of times when we're harvesting eyed larvae, we will grade them, and those larvae that we know are going to set within the next day, we'll catch on like a 180, and we'll put those in one tank. And those that go through the 180, we know we got 2 more days till we got to worry about those. We still drain them every day, but we know we don't have to screen them the next day.
Premixing your algae.
Yeah.
30 parts per 1,000 on the salt.
Yes. 30 grams per
why
liter.
30?
Good question. That was a recommendation from Reed Mariculture, and I'm assuming it has to do with the salinity that they raise their algae. But that's a good question. I'll ask them.
So you used to use fresh water to keep it from clogging years ago.
Yeah. The only thing I worry about fresh water is bursting the cells. You got a lot of osmotic difference there, and they may swell up with the fresh water.
Since we're talking about we do use that for supplement the nursery culture of clams. Do you know of anybody that actually uses it in larval rearing of hard clams? And what the success may be? Any idea?
I've never heard of any. Okay, sorry. All right. The question was, does anybody use shellfish diet, the algae concentrate, for rearing hard clams? I don't know of anybody. I would love to try it. We can't raise hard clams in Alabama. Our salinity is just not stable enough. But I would love to try it. I think it would work fine. I don't see why it wouldn't. The only question might be is the cell size. Is there enough of the smaller cell size for clams? I don't know if they eat smaller algae or not. Do any of our hard clam folks have input about algae size?
Sure. I think it's any different.
Yeah. So, in theory, it should work.
I also have a comment about the fresh water. I used to use the shellfish diet for eating freshwater mussels.
Yeah.
I would track it under a microscope, and it did not burst.
Yeah.
Freshwater, so.
Okay. Well, I'll follow up with Reed Mariculture on that and see if they have any input. The 30 parts per 1,000 was their recommendation.
Now you say you only had luck with the shellfish dye. You tried isoprices?
Well, we've tried. We've done live algae before, and it works fine. I'm sorry. The question was, have we tried other things besides Reed Mariculture shellfish diet? We've tried other pastes and other dried formulas, and the Reed product comes out, it's still fairly liquid. I've tried some pastes that were just thick clumps of algae and just smelled rotten, and they didn't work. I've tried some powders, they didn't work.
Well, the question was, you said you tried the isocrisis and the nano, and
Yeah.
you didn't have much success with those.
We tried different formulations. Nano does not work well for oysters anyway, but we did try higher mixtures with isocrisis and pavlova to try to get a lot more of those small cells, and we couldn't improve on their mixture. So why would you give yourself the headache of mixing your own when what they mix is really good?
Have you used INVE? Somebody's asking.
I have not. The question was, have I used INVE?
Or somebody suggested innovative aquaculture. That might be the same thing.
Innovative aquaculture. It's a product I'm not aware of. I'd be happy to try it.
Are there other questions from folks? Just taking nothing.
Scroll through here. You control water. How do you control water temperature during a larval stage?
Okay, so the question is, how do we control water temperature during the larval stages? Up until recently, we're an open air hatchery. So our hatchery is underneath our laboratory. It's fairly sheltered, but there are no sides. It is open air. So early in the season, in May, and late in the season in October, we do have some cool nighttime temperatures. We use a Finex titanium 500 watt heater. We hang it right in the airlift of the tank, and as that water passes, it heats up. That with a cover over the tank. We'll put a plastic cover over the tank if it's really cool, and that maintains temperature. We try to maintain 25 during the cooler parts of the year, and seems to work fine. Originally, when we started using it, I was really worried about cooking the larvae as they pass by it, but we haven't seen any mortality issues using heaters. Recently, I'll tell you this, going forward, we have enclosed part of our hatchery. We've enclosed one section to put in some broodstock conditioning systems. Before the commercial industry came to us and said, hey, can y'all back out? We were thinking about how are we going to meet the demand? Because we met all our orders, but we could have sold twice as many seed as we produced this year. So we're thinking of ways we can increase production. So we put in broodstock conditioning systems. We're also putting in some flow-through high-density systems that we're going to start working with. We've never worked with flow-through systems before. Batch culture has always worked really well. I've heard a variety of reports on flow-through systems. They reportedly can produce a lot more larvae than we can do in one of our big tanks in a much smaller footprint. So we've got a footprint of flow-through tanks that is probably 1/3 of what we've got, probably even less than that, probably 1/4 of the area we've got dedicated to batch culture. And we should be able to do as many larvae in that smaller footprint in flow-through tanks as we can do in our whole hatchery, the whole rest of our batch culture hatchery. It's something we haven't done before, but it is something we're going to try in the next year. Most of those systems run on cultured algae also. We are culturing a little bit of algae, or we're going to be culturing a little bit of algae, mainly to train our graduate students. But it's not going to be enough to run a flow-through system. So we're also going to be experimenting using the shellfish diet with a flow-through system.
Sorry to monopolize the question.
That's fine. That's fine.
Is there a significant time difference in getting the oysters high if you have low salinity versus high salinity?
Generally, if we're above 10 or 12 parts per thousand, everything seems to work pretty well. If we're closer to the 10 and under, it gets a little dicey. They really slow down. Yeah.
Somebody's asking if you produce your own tetraploids, or that's a 4C's deal. Or...
Okay. The question is about tetraploids. Do we produce our own tetraploids? Right now, all our tetraploids that we use to produce triploids come from LSU and 4Cs. LSU has an agreement with 4Cs. They produce the first line of tetraploids in the Gulf of Mexico. Currently, we are working with 4Cs on a line of tetraploids for Florida using Florida stocks. So we have tetraploids, we maintain tetraploids, and we are working on a tetraploid line for Florida. But we are licensed through 4Cs for using. But we have a flow cytometer, so we're able to check our own tetraploids to make sure they're tetraploids. That is one thing. If you're using tetraploids, make sure they're either checked before they come to you, or you have the ability to check them at your site. I've told the story a couple of times today. I did the first spawn of the season this year. We did a triploid spawn, and not sure exactly where we made the mistake, but it was likely one of our tetraploids that we had checked was diploid. Somewhere we made a mistake and I think we used 6 males, and one of those happened to be a diploid, and we ended up with a 2/3 triploid batch of larvae. We couldn't sell them as triploid, but there were plenty of buyers that were willing to pay a diploid price for 2/3 triploid.
You may have already asked this, but have you ever compared the shellfish diet, formulated diet to live algae?
All right, the question is, have we compared shellfish diet to live algae? It's a tough comparison. We've made some attempts at it, but it's really tough to produce the same algae at the same mixture. It's really hard to do. What I can tell you is shellfish diet works, and it works to produce a lot of seed. Is it better than live algae? I'd never say it was better than live algae.
It's more consistent.
Yeah. And you can keep a lot of it in your refrigerator. You know, they're always willing to send more. If your algae crashes, you can't grow more overnight. It's really worked well for us, and we have tried to make the comparison. It's just a very difficult comparison to make, to get everything in the right proportion. Live algae works great if you got it.
A couple more, sorry. How long do you run the hatchery each year? And yeah, that one first.
Okay, so how long do we run the hatchery each year? We typically start in May, around the 1st of May. For years, I tried to push it back into April, and anytime I tried to push it back into April, the spawns just didn't work well. So we start right around the 1st of May. Typically, we try to be done with spawning oysters and raising larvae by September. Done spawning mid-September, done with larvae by the end of September. The past few years, we've had to run longer to try to meet the needs that are out there. Last year, we ran larvae through October, and we ran seed through December. This year, we were thrilled. We produced a lot more seed, and we were done by mid-September with larvae, and we had all the seed out of the hatchery by Thanksgiving. Graduate students were thrilled. All right. Well, thank you all. Thank you, folks that are online.
Yeah, a lot of great practical information. Now, a lot was thrown at you. Remember, this is videotape, be posted by the end of the week, right, Carter? On the Shellfish Extension website, look under resources there. So I think you deserve at least a hand. Thank you so much.
Thank you. On the handout, my contact information and Bill Walton's contact information. Great resource for off-bottom aquaculture, for gear, for site selection, that kind of stuff. If you got any hatchery questions, feel free to email me or call me.